Hello guys, welcome to my channel. This is Isaac Simple and Real. Thank you for tuning in to my channel. Kindly, if you have not yet subscribed to my channel, kindly subscribe, like and share. This is a channel where we usually talk about issues concerning averting HIV and sexually transmitted infection stigma in our society. And we usually say that being positive about HIV is not necessarily being HIV positive. Welcome. Today we'll be discussing something different from what we have been discussing, but it's all about HIV. It's all about how to live safe, free, and also to live with HIV safely without infecting others, and also how to live without the virus, and also to avoid transmitting the virus to other people who are yet to be infected. Are we together? Thank you. Now, I have some demonstrations. I believe you can see clearly. Yeah. These are the demonstrations. And for these demonstrations, I'll be trying to explain how we're supposed to, or what is the life cycle of HIV, or how does one acquire HIV, how it progresses to replicating in someone's body and going ahead to infect another person. Are we together? Thank you. Now, uh, as you can see, this one is the CD4 T lymphocyte cell. It is usually available in the human blood system. It's one of the white blood cells that are available in the humans. And then on the other side, we have the HIV virus. Are we together? Thank you. Now, for the HIV virus to enter into my body or into your body or into any other person's body, it has to get through the CD4 T lymphocyte cell. Usually, when you have unprotected sex or through any port of entry with an infected person or person who is non-suppressed of HIV virus, usually this is what happens. I believe you can see it. This is the HIV virus, and this is the CD4 T lymphocyte cell. Usually what happens is you can see this virus, it has some projections. These projections, we usually call them HIV virus glycoproteins. Are we together up to this point? And these HIV virus glycoproteins are usually recepted or are usually received comfortably by the CD4 cell receptor. As you can see, there is a hoster or there is a space here, usually where the HIV virus glycoprotein invaginates into the CD4 receptor. Now, when the CD4 receptor receives the HIV virus glycoprotein, when it enters, the virus enters into the human body, the after invagination, the virus releases the genetic materials. In this case, HIV virus is an RNA virus, what we call a ribonucleic acid virus. Are we together? Now, when it invaginates into the CD4 receptor, it usually releases the RNA strands. Are we together up to this point? Now, this cell or this CD4 T lymphocyte cell it has received the HIV virus through the HIV virus glycoprotein, which has projected inside the CD4 receptor, and it has released the RNA strands. Now, these RNA strands proceeds to phase 2. Now, this phase 2 is what we call a reverse transcription. Are we together? Now, we had the RNA or ribonucleic acid, but now when we have reverse transcription, we have the virus releases an enzyme which we call reverse transcriptase enzyme. This enzyme converts the RNA strands into a double-stranded genetic material called DNA or deoxyribonucleic acid. So here we had an RNA or a ribonucleic acid, but here we have a deoxyribonucleic acid or a DNA, double-stranded DNA. Now, what happens? Now previously we had a RNA HIV, but now we have a DNA HIV. The next phase that usually happens within the cell, everything is happening within the CD4 T lymphocyte cell. The next thing that happens within the same cell is this infected cell, the virus inside produces another enzyme, which we call enzyme integrase. Are we together up to this point? Thank you. Now, this enzyme integrase integrates the human cell or the host cell DNA with the viral cell DNA. So, when the viral cell DNA is integrated, in this case, it appears like it has been hidden so that other antibodies cannot be able to detect there is a virus DNA here and there is a host cell DNA here. And in this case is where we call it a provirus DNA. Are we together? Thank you. Now, from this third phase, we go to the fourth phase. The fourth phase involves, now we had a DNA virus or a DNA HIV virus. We go to the fourth phase, which we call transcription. Now, this DNA virus in the host cell is usually activated through some enzyme that is usually produced within the host cell, which we call RNA polymerase enzyme. Are we together? Now, the RNA polymerase enzyme converts the double-stranded DNA HIV strand into so many messenger RNA or mRNA. Are we together? Now, this messenger RNA usually forms a long chain of viral proteins. Are we together? Now, this mRNA form a very long chain of viral proteins. Now, we go to the fifth phase, which is usually the assembly point. And in the assembly point, remember, we have the RNA viral strands. Now, these RNA viral strands combine with the virus protein. And this viral protein, remember they were a very long chain of proteins, but they are broken down using protease enzyme that is usually produced by the virus. Now, this virus produces the protease enzyme which breaks the long chain virus protein into short strands proteins. These short strand proteins, together with RNA, combine to form a virus particle. Are we together? Thank you. Now, we have already formed virus particle. And remember, these are very many RNA strands that are within this host cell and very many virus proteins that have been cut from the long chain virus protein. So we form a lot of virus particles. Now, these virus particles, they go to the sixth phase, which we call budding. Budding of Budding. Now, in the budding phase, this cell, as you can see, it is having some projections. This one, these projections. This is what we call budding. These virus particles, they form some buds. And because every cell has its lifespan, when this cell eventually ruptures, they will obtain cell materials, these cell membranes, to form the virus envelope. This is the virus envelope, which has been formed from the host cell membrane. And the virus, the virus proteins forms the projections of glycoproteins. And then the RNA is hosted inside. And we have a new cell of HIV virus. This is how this cell replicates. Then this cell, after it has been released, will go and attack another CD4 T lymphocyte cell, and the cycle continues. So, depending on the rate at which these viruses are replicating, it determines the prognosis or the progress or the initiation of first symptom of this person or this new individual who has been infected. So the window period can last from months to years, depending on who has been infected. Are we together? Also, it considers other factors, including the immunity status and also the rate at which the virus replicates. Are we together? Thank you. So kindly, if you have not yet subscribed to my channel, this is my appeal. Kindly, if you are watching this video, kindly subscribe, like, and share. This is a channel where I usually empower people to avert the issue of stigma, and also we encourage more people to learn how to live safely with HIV without infecting others, and also those who have not yet acquired the virus to prevent themselves from acquiring the virus. This is Isaac, simple and real. Bye bye and God bless you. Thank you.